Good evening, folks. I hope everybody is doing well tonight. This is part 2 in my lecture series on the fundamentals of DNA. I posted the first lecture in this series the other day. If you have not watched it yet, I recommend you go back and do that so you have some background on the DNA molecule going into our discussion here. If you were in my class, my biology class, I recommend that you go on the Blackboard site and download the handout that goes with this material so you have it to reference as the lesson proceeds. So we left off last time having detailed the history of DNA research, beginning from the discovery of the molecule by Friedrich Miescher in 1869 till the explanation of its effects with the work of Rosalind Franklin, James Watson, and Francis Crick in 1953. So along the way with that, we also learned a couple of important things about DNA. We learned about the 5' to 3' directionality of its strands. We learned about the principle of Chargaff's rules, whereby certain nucleotides are found in certain proportions in the DNA molecule. And we learned about the nucleotide itself as the basic structural unit of DNA. All things that were discovered on the way to Watson, Crick, and Franklin's final findings on the nature of the molecule structure. So, picking up from here, I want to start exploring the structure of DNA in more detail. What was actually found in 1953? If we learned all these other things along the way, what was the big discovery? What was found in 1953 was the structure of the DNA molecule. And knowing the structure of this molecule is essential to understanding how it does the things that it does. Now, when Watson and Crick began assembling their model, they initially thought that DNA was a 3-stranded molecule with the nitrogenous bases A, T, C, and G on the outside, and the phosphodiester bonds holding the phosphate and sugar backbone of the molecule on the inside. Turns out, flipped the other way around, that the phosphate sugar backbone of the molecule, made out of phosphates and deoxyribose sugars, are on the outside, and the nitrogenous bases A, T, C, and G point towards the inside. They were also wrong about the number of strands in the molecule. It turns out that DNA is not a 3-polymer molecule, but a 2-polymer molecule. That DNA consists of 2 separate chains of nucleotides wound around one another in a spiral fashion, like a spiral staircase. James Watson famously named that shape the double helix. And here is an image of the basic double helix of DNA. Now I've labeled it at top and bottom. Pardon me, my lamp is getting in the way here. There we are. Okay. Now I've labeled the double helix top and bottom. And you see I've got 5' to 3' on one of the 2 strands, 3' to 5' on the other one. Remember that DNA is always assembled in the 5' to 3' direction, but the 2 strands are running in opposite directions. In other words, one of them has its quote-unquote top, the 5' end at one end, the other one has its top at the other end. That running in 2 different directions structure is referred to as anti-parallel configuration or anti-parallel structure. So the 2 strands are wrapped around one another in this spiral shape. But look in the middle. There you can see the nitrogenous bases sticking towards the center of the DNA molecule. And now we can begin to understand something that Chargaff didn't fully get when he worked out his rules. He knew that whatever organism's DNA he looked at, the amount of adenine in it always equaled the amount of thymine, and the amount of guanine always equaled the amount of cytosine. Now you can see why. Adenine and thymine, and guanine and cytosine, are 2 pairs of nucleobases called complementary bases. Look at the word complementary here, spelled C-O-M-P-L-E-M-E-N-T-A-R-Y. Careful with that spelling. It is complementary with an E in the middle, not complementary with an I in the middle. Complementary with an I in the middle means that you get it for free, like the complimentary bag of nuts you get on an airplane. Complementary with an E in the middle, when 2 things complement each other, that means that they go together. And what Watson, Crick, and Franklin discovered is that there are two sets of complementary bases in DNA. A and T go together, G and C go together. And if you think back to the chemistry I told you last time, that means that you have one purine, that is one double ring molecule, and one pyrimidine, one single ring molecule in each of those pairs. Now, when I say they go together, looking back at our DNA molecule over here, you can see that what's happening is that the nitrogenous bases reach across the middle of the molecule and form hydrogen bonds with each other. The two strands of DNA are actually held together by a series of hydrogen bonds between bases on opposite sides. However, they can only form hydrogen bonds in two specific ways, and you've probably already guessed what those are. Adenine cannot form a hydrogen bond with guanine, cytosine, or another adenine. It can only form a hydrogen bond with thymine. Their shapes complement one another and allow that. Actually, adenine and thymine form two hydrogen bonds between them whenever they come together. Likewise, guanine and cytosine match each other, they complement each other, they can form hydrogen bonds with one another, but neither of them can form a bond with itself or with adenine or thymine. Cytosine has to bond with guanine, the two of them form three hydrogen bonds with each other. So in other words, the two strands of DNA are held together across the middle by a series of hydrogen bonds between complementary bases. So wherever there's an A in one strand, as you can see, there's got to be a T exactly opposite it in the other strand. Wherever there's a C in one, there has to be a G opposite it in the other, and vice versa. Those complementary bases are what's holding the molecule together. Each pair that's formed in that way, incidentally, is called a base pair, or BP for short. Base pairs are the fundamental units by which we measure the length of a DNA strand, or the length of a gene. We'll say, okay, this gene is 446 base pairs long, or this DNA strand is 1.2 million base pairs long, or whatever. Human DNA, if you get all the human DNA together from all of our chromosomes, is a total of about 5 billion, billion with a B, base pairs long. So that is the overall structure of the DNA molecule. Two anti-parallel strands running opposite each other, 5' end of one strand at the top, 5' end of the other strand at the bottom. Held together across the middle by nitrogenous bases that stick towards each other in the middle. They form hydrogen bonds with each other. A and T are complementary bases, form two hydrogen bonds between them. C and G are complementary bases, form three hydrogen bonds between them. And those hydrogen bonds hold the two strands of DNA together. Now, this structure is important for a couple of reasons. For one, it is incredibly strong. An individual hydrogen bond, as you all know, is weak. But you've got two of them between every AT pair, you've got three of them between every GC pair, and you have how many base pairs in your DNA? 5 billion? That's a lot of hydrogen bonds pulling in the same direction. And even though individually they are weak, together they are incredibly strong. This gives the DNA a lot of structural integrity against the possibility of being damaged by outside forces. And considering your DNA is your cell's genetic record of all the instructions on how it works, keeping that intact is super important. Another reason this is important is because it is easy to repair if it does get damaged. Sometimes DNA will be damaged by outside forces. But remember, if you know the structure of one strand, you automatically know the structure of the other. If one strand goes ATCG, the other one has to go TAGC, because A always pairs with T, C always pairs with G. So let's suppose my DNA does get damaged, and part of one strand is damaged. Well, guess what? As long as the other side is intact, we can rebuild the damaged part exactly the way it was before. Because even if I knocked off a few nucleotides with some outside chemical damage or whatever, anytime I knocked away a T, if there's an A on the other side, only a T will bond with it. If there's a G on the other side, only a C will bond with it. So in other words, as long as one strand is intact, you can rebuild the other strand from it exactly the way it was before. This structure gives the DNA molecule a high degree of fidelity in repairing damage. And it also incidentally makes things easier for us as biologists, because if I know the sequence of one strand, I automatically know the sequence of the other strand. And finally, the structure of DNA makes it remarkably efficient to replicate. Think back to whenever cells divide. In the interphase leading up to cell division, you have the S phase. During the S phase, remember, you have to duplicate all of your chromosomes. And I'm duplicating my chromosomes. That means I am copying all of my DNA. It is vitally important when cells divide that every cell is getting a complete copy of the genetic information, that it's got all the genes that it needs to function, and it's equally important that those genes be assembled properly. In other words, that not only do we get all the genes, but they look like the ones that we had previously. Mess with the instructions, it's like messing with an old cake recipe. Grandma's old cake recipe is not going to come out the same way if you change the ingredients. So, we want a way of replicating this molecule that is highly faithful, that ensures that the code in the original molecule will be the same as the code in the new DNA molecules that we form during S phase. Now, when I'm thinking about a molecule that is 5 billion base pairs long, you might think, well, that's a lot to get right and copy faithfully. But the structure of the DNA actually makes that easier than you might think. During the process of DNA replication, 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 when this thing is replicated, it's copied. During the process of DNA replication, what's going to happen is that the two strands of the DNA are going to unwind. They're going to come apart from each other. We often say that the DNA is being unzipped like that when that happens, like we're opening a zipper. The two strands separate from each other. Then we are going to build a new complementary strand, like complementary bases, onto each of the old ones. So we're going to pull the two strands apart, and we're going to build a new complementary strand for each of them. But remember, A has to pair with T, C has to pair with G. Chargaff's rules tell us that that's the way it has to work. What that means is that the new strand I build will be exactly like the old one that I pulled away. So following that process, whereby we unzip the strands and assemble a new one on each of them, what we should have is two complete new DNA double helices, each one identical to the preceding one. So that complementary base pairing structure ensures faithful copying of the molecule. This is not to say things can't go wrong. There are things that go wrong. We'll talk about that later. But in general, that process makes the DNA molecule highly replicable and able to do so in a faithful way that preserves its genetic information. That said, kind of like with cell division, remember that whenever we talked about mother cells versus daughter cells, that whenever I have two new cells at the end of mitosis, it's not like one is the original and one is the copy. Both of them are new daughter cells because the original got ripped in half during the process. Same with DNA. The process of DNA replication is often called semi-conservative. And what we mean by that is that the data in the DNA, the sequence of nucleotides, is conserved. That means it's going to stay, it's going to still be there at the end. Both molecules, assuming all went the way it should, are going to have the same genetic sequence at the end as they did in the beginning. The information is conserved. The molecule itself is not. Because during DNA replication, we ripped the two strands apart. We destroyed the original molecule, and then we built a new strand onto each of the old ones. So DNA is semi-conservative. We conserve the information when we replicate it, but we tear the molecule apart and use those pieces to build new molecules in the process. So this base pairing structure of DNA allows us to form complementary strands that are duplicates of the ones that were there before. Structure of DNA makes it highly easy to protect, easy to repair, and easy to copy. So this is all quite important for understanding how it works. Sound good? Okay, that's the structural stuff. Let's talk function. There are two big functional aspects of DNA that we need to discuss. The first and most obvious of those is how it codes information. how is it that that series of A's and T's and C's and G's is actually providing instructions on how to make the color of your eyes and the shape of your nose? The other thing we have to consider is, how do we replicate it? I told you that we unzip it and build new strands onto the old one, but that is a heavily simplified version of a very complex set of processes that have to happen quite faithfully and quite quickly. So we need to talk about two big things about DNA: how it encodes data and how it is replicated. We're actually going to begin with the second one of those, because I think it's a lot more important, at least initially, to know how we build DNA before we can understand how it works. So we're going to begin with DNA replication. Now, at its simplest level, DNA replication just means we are going to make two new DNA molecules from one original. Unzip the strands, build a new complementary strand onto each, and now we have two copies of what we started with. DNA replication happens during the S phase of the cell cycle. This is the part of the cell cycle prior to actual mitosis, prior to G2 actually, when the chromosomes are copied. Remember that I said that one of the things that happens during that is the chromosomes condense and become visible during mitosis. The reason they have to do that is because before mitosis, they weren't condensed. The DNA was all loose. It had to be unpacked from the chromosomes so that we could pull the strands apart and copy it. So S phase of the cell cycle is when this happens. The DNA loosens up from the histones in the chromosomes, from the packaging proteins, so that it can be duplicated. How does that work? Before I can tell you about the duplication process itself, let's introduce some of the enzymes involved. DNA replication is a very complex process with many distinct chemical steps. It's a very involved biochemical pathway. And like all biochemical pathways, it's going to benefit from a lot of enzymes and coenzyme proteins that help out along the way. So I want to introduce you, before we get started, to the various enzymes and accessory proteins that are going to be involved in this process of DNA replication, give you sort of an enzyme language to speak up front. Now, the enzymes involved in DNA replication fall into three basic categories. First and foremost, we have the polymerases. Polymerases are DNA building enzymes. These are the enzymes that actually assemble new DNA. Polymerases always work in the 5' to 3' direction. In other words, they always assemble new DNA strands by attaching the 5' phosphate of one strand to the 3' hydroxyl of the preceding strand. Now, there are about a half dozen known DNA polymerases. The two of them that are most important are the two I have listed here: DNA Pol 3 with Roman numeral 3, and DNA polymerase alpha with the Greek letter alpha. Find that in the insert symbol part of your word processor. DNA Pol 3 is the primary polymerase in prokaryotes. If you're a prokaryote, this is the main DNA building enzyme you use. DNA polymerase alpha is the primary polymerase in eukaryotes, in our cells. This is the main polymerase we are using. Those are the only two polymerases I really care about you knowing about. Take a genetics class, you'll learn about some more. Now, polymerases always work in the 5' to 3' direction, assembling new DNA. And for all the reasons I've outlined, mistakes are rare. But sometimes errors do creep in. When you're copying out 5 billion base pairs, odds are something is going to go wrong slightly along the way. Therefore, DNA has what we call a proofreading function. The proofreading function of DNA is its ability to check itself for errors to see if something went wrong during replication. The proofreading function is carried by a group of enzymes that we call the nucleases. Wait, too far, here we go. All right, my nucleases. Nucleases are the proofreading enzymes of DNA. They are the ones that check for errors. Nucleases typically work in the opposite direction from polymerases, 3' to 5'. There are basically two ways that nucleases do their job. We talk about different nuclease functions. These are not names of enzymes. These are two ways that nucleases can operate. First off, we have an exonuclease function. E-X-O-N-U-C-L-E-A-S-E, exonuclease. The exonuclease function means that the nuclease will eliminate errors in the DNA starting from the end of the strand and removing every nucleotide between the end of the strand and the point of error. I drew a sample DNA strand here. You can see we go A, T, C, G, G, A, T, C, A. I've circled the second G because that one we're going to say in this sequence is the error. That's the one that should not have been there. You can see that in our exonuclease, we started from the A at the very end, and we eliminated everything from the end of the strand all the way up to the erroneous nucleotide. That's an exonuclease function. Then we have an endonuclease function, E-N-D-O nuclease. In an endonuclease function, we don't take out the whole strand, we just take out the one bad nucleotide. So in our endonuclease function, we don't have to eliminate the 4 at the end to get at that 1 in the middle. We can just pluck out that one bad G. You might reasonably ask yourself, okay, so why don't we just always use endonucleases? If that's the precision way that just plucks out the one bad nucleotide, why isn't that the preferred method? The answer to that is mutations are not always that simple, folks. Sometimes you're going to mess up a whole segment of DNA at once, in which case it's much more effective to take out a big swath of it at one time than it is to take out every individual nucleotide that is in error. We're going to talk in the next chapter about mutation and find out about different kinds of mutations, some of which you're going to see are more radical than others. Actually, 2 chapters from now, not the next chapter. Let's hit RNA first. Now, I didn't list specific nuclease enzymes for you. I showed you nuclease functions. And the reason for that is because it turns out that nucleases are not going to be specific enzymes that are unique in their own regard. Most nucleases, it turns out, are actually polymerases. They are polymerase enzymes working backwards along the same strand. It's a rare example of an enzyme doing 2 different jobs. The polymerases will build the DNA in the 5' to 3' direction, and then they will work backwards in the 3' to 5' direction, carrying out a proofreading nuclease function. There are a couple of specific nuclease enzymes, but again, we're not going to get into how they work differently. In general, think of nuclease activity as a secondary function of polymerases. And finally, we're going to have a large group of enzymes that we collectively call the accessory proteins. The accessory proteins are various enzymes and proteins that carry out, you might say, miscellaneous functions in the process of DNA replication. They're assisting with various aspects of this thing that do not necessarily involve directly building or editing the DNA sequence, but things that need to happen along the way. Now, there are dozens of different accessory proteins. I have listed for you here some of the most important ones that are likely to come up in our discussion. We have DNA primase, P-R-I-M-A-S-E. DNA primase is an enzyme responsible for building a little support molecule known as the primer. Now remember how I said that DNA is always built in the 5' to 3' direction. In other words, that we join the 5' phosphate of one nucleotide to the 3' hydroxyl of the preceding nucleotide. That leaves us with the question of what do you attach the first nucleotide to? If you always have to hang it on the one that came before, how do you put the first one in place? The answer to that is that you attach it to a tiny molecule called the primer. DNA primase builds a short segment of RNA called the primer. The RNA primer serves as the anchoring point for the first nucleotide in the DNA sequence. So we need a primer before we can begin building new DNA, and DNA primase assembles that. Now, once you've gotten that started, you don't need the primer anymore. The primer can then be removed with the help of this enzyme right here, DNA Pol-1. DNA Pol-1's job is to remove the primer once the new DNA sequence is started. We also have DNA helicase, H-E-L-I-C-A-S-E. DNA helicase is an enzyme that unzips the DNA strands. DNA helicase breaks the hydrogen bonds between complementary bases and pulls the 2 strands apart. It is gradually active during the process of replication. Separating the strands does not happen all at once. It is a slow, ongoing process during replication. So DNA helicase is continually moving down the molecule, separating the two strands as the process unfolds. Now, as you separate the strands, we have the very real risk of these strands, which have a natural tendency to coil and kink, getting kinked or twisted during the process. That is where DNA gyrase comes in. DNA gyrase is responsible for making sure that they do not become twisted or kinked along the way. DNA Pol-1 we already mentioned. DNA Pol-2 is a repair enzyme. It's not responsible for building new DNA. It's responsible for fixing damage. After a nuclease function finds an error, DNA Pol-2 will go in to repair it. Then we have DNA ligase. DNA ligase has a rather unusual function. It is responsible for joining together disconnected fragments of DNA that are called Okazaki fragments. You capitalize the O in Okazaki, named for the Japanese scientist who first described them. So DNA ligase has the function of joining up Okazaki fragments during DNA replication. Okazaki fragments are small, disconnected bits of DNA that form during the replication process. We will tell you more about what they are and why and how they form in a few minutes here. And last but not least, in our list of accessory proteins, we have SSB, lowercase letters for that, SSB. SSB stands for single strand binding protein. The purpose of SSB is to cover up hydrophobic regions of the DNA molecule during the replication process. Certain portions of the DNA molecule are hydrophobic, and you guys know that hydrophobic exclusion means that organic molecules tend to form shapes where their hydrophobic parts are tucked away on the inside of the molecule. DNA is no exception. But here's the thing. Once you unzip the two strands, those hydrophobic regions in the interior of the molecule are now exposed to the watery environment of the cell. And you all know what that means. Hydrophobic exclusion is going to try to make the molecule bury those parts, which is going to cause the thing to bend and fold in ways that we do not want it to be doing. So SSB is a mask of sorts. It covers up the hydrophobic regions while the DNA is unzipped to prevent the molecule from trying to shape itself to avoid contact with the water. So these are just some examples of accessory proteins. This is not an all-encompassing list of them, but they're the ones that are probably most important to the process that we are going to find coming up the most frequently along the way. By the way, you might have noticed that two of those accessory proteins were called Pol-1 and Pol-2, Pol-1, Pol-2, and that one of our polymerases was DNA Pol-3. The reason for this is that whenever these enzymes were first discovered, back when they were first being discovered, we didn't yet know which ones were involved in DNA building versus other activities. And if you think about what Pol-1 is doing, it's building a little segment of RNA, which makes it look like it's a DNA assembling enzyme if you don't pay too close attention at first, or if you don't know what's going on at first. Likewise, Pol-2 is a repair enzyme. So if you're watching its activity out of context, it also looks like it's helping to build DNA. So they were named polymerases when they were first found. And all the polymerases were numbered in order of discovery. So DNA Pol-1 was found first, Pol-2 was found second, Pol-3 was found next. But eventually we discovered that Pol-1 and Pol-2 weren't actually polymerases at all. They were not DNA builders. They were carrying out other activities. However, the names stuck, which is why we have DNA Pol-1 and 2 as accessory proteins, but DNA Pol-3 as an example of a polymerase. All right, sound good? Okay, so that is kind of our enzymatic cast of characters. And I know that's a lengthy list of enzymes and proteins that are going to come up along the way during our process of DNA replication. Okay, so now let's find out what replication is actually like. The process of DNA replication has 3 distinct steps. Step 1 is initiation, the preparation phase. Step 2 is elongation, the building phase. And step 3 is termination, the finishing phase. Each of these is its own set of biochemical pathways carrying out a variety of different actions to copy the DNA molecule. Now, we begin with initiation. Initiation is our first initial step. This is our prep step, in which we are getting the molecule ready for the process of replication. Now, interesting thing about this, we talk about unzipping the DNA molecule. In reality, that's kind of a poor description, because unzipping implies we're opening it up from one end towards the other end, like pulling a zipper. It's not quite how it's going to work. A better way to think of it would be to imagine you're splitting a zipper apart in the middle. You know how a zipper sometimes splits, it starts opening out towards both ends, and the teeth of the zipper are all apart, and you're trying to shove it back together so you can properly unzip it, and it's a real mess. That is how DNA replication proceeds, that rather than starting at one end of the molecule and working towards the other end, we are instead going to start the process of replication in the middle of the molecule. And we are going to work bidirectionally out towards both ends at a time. See what's going on there? So those are our two strands of DNA. I can't really draw them well in 3 dimensions. Let's just pretend that I've just drawn a beautiful, elaborate spiral of DNA wrapped around one another in a 3D helix, and you are super impressed with my artistic abilities. But let's just say that that's what's happening, but for ease of your understanding, of course, I drew these 2 parallel lines. These are the 2 strands of DNA. I've omitted the nitrogenous bases and things here just to simplify what we're looking at. But our replication process starts from part way down the length of the strand, somewhere around the middle, and rather than unzipping from one end to the other, we're going to unzip from the middle out in 2 directions at the same time, like a zipper splitting apart. So it's going to kind of bulge in the middle and bulge out in the center, and that bulge is going to open up more and more towards either end as the process goes on. So the DNA replication process proceeds bidirectionally from the center. Now, in the middle of that DNA sequence, not every letter of the DNA is necessarily coding for genetic information. Some of those A's, T's, C's, and G's in there are carrying out other functions altogether. We're going to talk about what some of those are as we go along, but here's one right now. That around the middle of the DNA sequence, you're going to find a stretch of nucleotides that do not code for any genes at all. That little group of nucleotides is called the origin of replication, sometimes abbreviated ORI-C, capital O, little r, little I, capital C. The origin of replication does not code for information. It serves as a starting point for the replication process. This is where the process is going to begin. So here's how it's going to work. Step 1, initiation. Starting at the origin of replication, partway along the length of the strand, we are going to begin assembling all the stuff that is needed to copy the DNA. We're going to need to gather together all those various enzymes that I was telling you about before. So these enzymes are going to begin to gather at the origin of replication, which I represented in my diagram right here as a couple of little boxes, one on each side. That boxed in area is the origin of replication. The origin of replication, we're going to gather together these enzymes, and we are going to form 2 separate multi-enzyme complexes. Remember, a multi-enzyme complex is all the different enzymes needed for a particular biochemical pathway. So we're going to assemble 2 different multi-enzyme complexes that will attach to the DNA at the origin of replication. The first one we assemble is called the primosome, P-R-I-M-O-S-O-M-E. The primosome includes DNA primase, remember the one that builds the primer, DNA helicase, the one that unzips the strands, and a couple of miscellaneous accessory proteins that we didn't talk about. Build the primosome first. Then we build the replisome, R-E-P-L-I-S-O-M-E. The replisome is going to include all the other enzymes that we talked about, a couple that we didn't talk about, but most importantly, the replisome is going to contain 2 copies of whichever polymerase enzyme we need. So if you're a prokaryote, the replosome will contain 2 copies of DNA Pol III. If you are eukaryote, it'll contain 2 copies of DNA polymerase alpha. And the reason for that is because this process is going to proceed bidirectionally. We're going to be copying the DNA in 2 directions at once from the middle. And therefore, we need 1 polymerase for each way. So the replosome is our second thing assembled. We build the primosome first, and then the replosome. They both attach to the origin of replication. Once this happens, the primosome goes to work. The primosome is really the prep multi-enzyme complex. The replosome is the actual replication multi-enzyme complex. And so what happens here is that the primosome goes to work, which means that DNA helicase begins unzipping the strands from each other, pulling them apart, and DNA primase begins assembling the primer, that little segment of RNA that's going to serve as the starting point for building new DNA. So that is our process of initiation, the prep stage. To summarize everything that happened in initiation, we gathered together all of our enzymes at the origin of replication. We built 2 multi-enzyme complexes, first the primosome and then the replosome, which attached themselves to the DNA. Then we began unzipping the DNA with DNA helicase, and DNA primase assembled our primer. Both of those are actions of the primosome. That's initiation. That's step 1. So during step 1, our DNA starts off looking like that, 2 parallel strands that are still joined together. By the end of initiation, it's going to look more like that. You can see where it's bulging in the middle, that the strands have begun to pull apart from each other in 2 directions at once. And OREC, origin of replication, is that little boxed in part on each side. Okay, so that is initiation, prep stage. We're getting ready to go. Now we move to step 2, elongation. Elongation is the building stage. This is the longest stage of the process. This is the step during which we are actually going to assemble new DNA. Now, elongation is going to proceed in 2 directions at the same time. We're going to be going this way and that way out from the middle. So everything I'm about to describe to you in elongation actually happens twice. It's happening going in both directions at once. Now, during elongation, the primosome is going to steadily move along the length of the DNA molecule, with DNA helicase gradually unzipping the nucleotides from one another, one by one. So the DNA does not unzip all at once. It's a slow and gradual process with the strands pulling more and more apart from each other, working from the middle towards the end. And that's happening in both directions, so everything that was happening on that one side happens the other side as well, opening out that way. What this means is that at any given point in time, during the process of elongation, there's going to be a place where the strands are separated from each other, but there's also going to be a point where they're still joined together. We're going to have this sort of Y-shaped arrangement where the strands are partly spread apart, but there's also a point right in the middle where they're still connected. That Y-shaped spot that forms right there is called the replication fork, like a fork in the road where it goes 2 different ways. The replication fork is where the action happens. Because during the replication fork, not during the replication fork, because at the replication fork, that's the point where the strands have just pulled apart from each other. And that is always the point where you will find the replosome. The replosome is always found at that spot because the replosome stays there to build new DNA. As the strands pull more apart from each other, the replosome moves into the gap to assemble new DNA. Now, here I got for you a diagram of elongation. This is meant to show what's happening on one side of the molecule and one side of the molecule only. Everything that I'm showing you is happening in mirror image on the other side. Now remember, we've got 2 strands of DNA. In this diagram, the uppermost strand is the one that is oriented 5' to 3'. Remember, one of the strands is 5' to 3'. The other one is reversed, flipped the other way, 3' to 5' because of anti-parallel structure. Now remember, this is quite important. DNA can only be built in the 5' to 3' direction. You always have to assemble new DNA by linking the 5' phosphate on one nucleotide to the 3' hydroxyl on the preceding one. We laid our first nucleotide in place by attaching it to the primer. Then we can attach subsequent nucleotides one after another to the one before them. Once that has begun, DNA Pol 1 removes the primer. The primer is no longer needed. I didn't even bother drawing it in this diagram. Now take a look though. We got two strands of DNA going in two different directions. One is 5' to 3', one is 3' to 5'. The one on the top in my diagram is the 5' to 3' strand. It's going to be the easier one to build new DNA along. It's going to be faster. It's going to lead the process because 5' to 3' is the direction we need to go. For that reason, the 5' to 3' strand of the DNA is often called the leading strand. The 3' to 5' strand, however, is the opposite of the way we need to go. It's going to lag behind. It's going to be slower to build. So we're going to call it the lagging strand. Now here's what happens. During elongation, we're going to slowly unzip the strands from each other. DNA helicase is going to be gradually pulling them apart from each other during this whole process. As that happens, the replosome is going to move along the leading strand. The replosome is going to move along the length of the leading strand. It always stays at the replication fork. Whenever you separate more nucleotides from each other, it moves into the gap that formed. So it's always found right there at the juncture of the two strands. Replosome always moves in the 5' to 3' direction. As it does so, the polymerase within it is going to add new complementary bases. We are going to assemble a new strand of DNA alongside the old one. We follow the basic laws of complementarity here. Wherever there's an A in the original strand, you put a T in the new one, and vice versa. Wherever there is a G in the original strand, you put a C in the new one, and vice versa. So look at my upper strand there. My replosome has been making its way along the leading strand in the 5 to 3 direction. You can see there was a T as the first nucleotide in the sequence, so our new one, the replosome put in an A. There's a G as the next one, replosome put in a C. Next one is a C, replosome puts in a G, and so forth and so on. You can see that the replosome right now is at the replication fork, right at the point where the two strands are just separated from each other. The nucleotide right at that juncture is cytosine, C, so the replosome just put a G right in that spot. In a moment, DNA helicase is going to unzip the next pair. The replication fork is going to move further down the strand by a little bit. We see we've got a TA pairing there. So whenever the T gets pulled away from the original strand, replosome is going to put an A in for the new one. And in that way, we are going to gradually proceed along the length of the leading strand, building new DNA alongside it in a continuous sequence, one nucleotide at a time. So far, so good. That's the easy part. Here's where it gets harder. DNA can only be assembled in the 5' to 3' direction. But our strands do not both go 5' to 3', do they? The leading strand runs 5' to 3', and therefore it's easy to build along. The lagging strand is the opposite. We cannot build in the 3' to 5' direction. So here's what's going to happen. The lagging strand, as its name suggests, is going to lag behind. We're going to be building along the leading strand, but nothing's happening on the lagging strand just yet. But what is going to happen is that after a while, we're going to build some DNA along the leading strand. We're going to build anywhere from 28 to 30 nucleotides along the leading strand, assemble a new segment of DNA about that long. In my diagram, I didn't make it that long because I'm just trying to show you what's happening, and I didn't want to take time to draw 30 or 60 letters there. But we're going to assemble a section of DNA along the leading strand. Then, after 28 to 30 nucleotides, we're going to move backwards along the lagging strand. And we are going to assemble a small bit of DNA moving backwards along the lagging strand. So we're going to reverse direction. The replosome is going to work backwards. Because if I'm going backwards along the lagging strand, backwards is 5 to 3. I can build in the 5 to 3 direction. So I'm going to build a little chunk of DNA working backwards along the lagging strand, because that's 5 to 3. That's the direction we can build in. That little bit of DNA that we assemble is what we call an Okazaki fragment. So there's my Okazaki fragment that I talked about before. Same rules apply. Wherever there's a C in the original, we're going to put a G in the new one. Wherever there's a T in the original, we're going to put an A in the new one, and so forth. I didn't draw them in here, but you can see how it would go. And we are going to repeat this process throughout replication. We are going to build the leading strand in one long, continuous, unbroken sequence. And then the lagging strand is going to be built working backwards a bit at a time in a series of disconnected Okazaki fragments. Every 28 to 30 nucleotides along the leading strand, we're going to pause and work backwards and build a bit of lagging strand. As we are doing all of this, several things are happening. DNA gyrase is acting to keep the strands from becoming twisted along the way. SSB is covering up hydrophobic areas along the way to prevent the strand from trying to fold itself up as a result of hydrophobic exclusion. DNA gyrase is keeping things from getting twisted or kinked. Wait, I already said that, didn't I? And DNA ligase is joining the Okazaki fragments together. As we build an Okazaki fragment, we're going to connect it to the previous one. DNA ligase is going to bind them together. So at the end of the process, it ends up looking like one long continuous sequence, even though it was built in discontinuous bits. All right, so far so good. Now remember, everything that I showed you here is happening in mirror image going the other way. So the other side, remember, is going to be proceeding outward from the middle, going in both directions at once. I showed you one side. Everything happening here is happening in mirror image on the other side at the same time. The only difference is going the opposite way. We're going to reverse which one is leading and which one is lagging. Can you see why? Because going the other way, the leading strand will be going 3 to 5. Going the other way, the lagging strand will be going 5 to 3. So that is elongation. Little bit by little bit, we are unzipping the strands from each other, working our way towards the ends of the molecule. And little bit by little bits, we are building new DNA alongside each of those old strands. So that's elongation. Step 2. Now we get to the third and final step of DNA replication, which is termination. Now, termination is going to end up kind of like initiation. We had a starting point called the origin of replication, a sequence of nucleotides that doesn't code for anything. Its entire job is just to indicate here's where you begin. At either end of our sequence, we are going to have another similar stretch of nucleotides that does not code for anything that is called the termination site. Once our replosome hits the termination site and copies it, that signals to it, hey, we're done. At that point, the replosome stops building new DNA. Our polymerase enzymes then go back over the DNA we have just assembled, carrying out the proofreading function, checking for errors. Any errors that are found will be eliminated through either endonuclease or exonuclease activity, and then repaired with DNA Pol II. Once the DNA has been proofread and all errors discovered have been repaired, the two new DNA molecules pull apart from each other. The last thing that happens during termination is that there's a sort of little cap going to be added to each end of the molecule. Those caps found at the end of the molecules are known as telomeres. T-E-L-O-M-E-R-E. Prefix telo means at the end of, mere means segment or units. The telomere is basically just a string of nucleotides, usually just a repetitive sequence of cytosine nucleotides. Doesn't code for anything. The telomeres are added on to the sequence at the very end, and they serve the same function as the aglets on your shoelaces. If you don't know that word, there's your vocabulary word for the day, A-G-L-E-T. That's the little hard cap on the end of your shoelaces. The aglets of your shoelaces protect the delicate ends of the lace from becoming frayed or damaged. That is what telomeres are for as well. Telomeres protect the ends of the DNA molecules from becoming damaged or frayed. So we have the telomere as a cap on either end of the DNA in order to protect it. That's the last thing that happens during termination. So, at the end of termination, what we now have, assuming everything went the way it was supposed to, is 2 new DNA molecules, each one a perfect replica of the one that we had to start with. Assuming everything went the way it should, no mutations slipped through the cracks, we should now have 2 identical novel DNA molecules, each the same as the one we had at the beginning. And that, folks, is DNA replication. Now, the way I have described it is pretty universal. The process of DNA replication has very little variation from one living thing to another. The basic steps are strongly conserved in the evolutionary record. They date back to the earliest life on Earth. And as such, the replication process is pretty much the same in most organisms. That said, there are going to be a few little differences that we are going to encounter from one group of organisms to another, primarily differences between prokaryotes and eukaryotes. But I think I've given you all enough to chew on right now. That was quite an elaborate series of steps that we had to go through. So I think this is a good time to call it a night. So now you've learned how DNA is replicated. Next time we get together, we're going to close off this chapter. We're going to talk about a few differences in the replication process between prokaryotes and eukaryotes. We're going to talk about why telomeres are so important and what that has to do with aging. And we're going to learn about how exactly your DNA encodes biological information, and along the way, find out what this thing called a gene really is. Okay? Sound good? All right. Hope you all have a good evening. I'll talk to you soon. Good night.